simple cells versus what we're going to consider complex cells. We're going to add some more descriptive terms that separate those two. But now that we're in Unit 2, we're actually zooming out to the level of cells in the first place. We have been in the level of atoms and molecules in Unit 1. I think one of the last questions on the exam were listing those 4 major biomolecules, giving it examples of them. So that's where I'm at on the grading the exam stage. But today, as we jump into new stuff, we're going to talk about cells and organelles. And I wanted to run into lab and get our first model of a cell. We can see this is one of the big complex cells. It's got a bunch of internal compartments, and we're going to view a few of those today and see how far we can get. One of the things we're going to ask is, why are cells so small? And I'm kind of restricted on a 2-dimensional surface to draw this, but I'll do my best. What I don't like about this is I'm working on a 2-dimensional surface, and we're talking about a 3-dimensional cell. Let's just say this represents the boundary of a cell, our plasma membrane. This is the surface area that we have on a cell. And inside you've got this volume that would be filled with cytoplasm. What happens with cells is that as they start to grow larger and expand, their volume inside, the red area, is going to increase much more rapidly than the surface area would around the outside. So if this cell was to swell to a size this large, its surface area has increased, yes, but the volume is going to increase much more rapidly than this surface area does. So cells, if they get too large like this, they have passed this crucial threshold where now there's so much volume inside that the organelles are not going to be able to interact properly. And things like oxygen, sufficient amounts of oxygen, are not going to be able to diffuse all the way to the middle of the cell. And carbon dioxide that's produced inside the cell might not be able to diffuse sufficiently all the way out. So when cells get too large, it's hard to get proper amounts of nutrients in and waste out. Cells have to exist in this kind of below this threshold where there needs to be more surface area than there is volume. Cells that are real tiny like this, there's not a lot of volume in there. So there is, the smaller a cell is, the more surface area it has in relationship to its volume inside. Okay, here's a picture of 2 types of cells. The first one that's in the foreground is going to be what we refer to as one of these simple cells. We're going to list at least 4 or 5 differences between the types of cells. The first thing that we can see in our simple cell, it gets a name called prokaryotic cell. Let's put that up here. Simple cells are prokaryotes. And we can make that term make sense a little bit. Pro just means before, and karyote means nucleus. So prokaryotes are cells that don't have a nucleus. They were around before a true nucleus was. Here is a nucleus in our cell model. It contains all of the DNA. And then we have all these other compartments outside. Some cells, our simple cells, don't have a nucleus. They've got DNA right here, but it's not separated by any kind of membrane. It's just kind of floating around in the cytoplasm of the cell. We can also see some dots that are present in these simple cells as well as in these complex cells back here. Those dots are going to be called ribosomes. And we're going to learn that ribosomes are the assembly sites of proteins. I don't know if you want to write that down somewhere, but we will talk about the whole process of building proteins. We'll call it protein synthesis. And I'll describe it as a dance that takes place between multiple types of RNA. And like any dance, it needs to take place on a dance floor. So the ribosomes are the dance floor on which this dance of protein synthesis occurs. So whenever we see a ribosome, you can think proteins are being built anywhere there are one of those dots. All right. We can see another difference between these larger eukaryotic cells and the simple prokaryotic cells from this one picture, and that is that these eukaryotic cells are much larger. They don't even fit on the screen compared to this prokaryotic cell. So prokaryotes don't have a nucleus. We also said they have no internal compartments, no organelles. We said the complex cells we're going to call eukaryotes. Give myself a little bit of space there. Karyotes, again, refers to nucleus. And U in front of it means true. So eukaryotes are cells that have a true nucleus. In addition to the nucleus, we can see they have other membrane-bound organelles present. The other thing that we can see on there is size. Prokaryotic cells are small. I'll say smaller. All cells are small. Prokaryotic cells are much smaller compared to these complex cells, which are larger. Larger, they got a true nucleus, they got many internal compartments. Before I get too carried away with the differences between these two, let's list 4 similarities that both cells have in common. There are 4 similarities between all these cells. We're going to see whether it's a complex or a simple cell, they are all going to have a plasma membrane. They're all going to have cytoplasm, the fluid inside that plasma membrane. And then floating around in that fluid, they're going to have DNA that contains the instructions to build our proteins. And then we need a dance floor, a site to actually build those proteins, and that's where ribosomes come in. So all cells, bless you, are going to have those same 4 characteristics. Even on this simple cell, there is just underneath this exterior, this cell wall, there is a thin plasma membrane. It's the red layer. And then inside that, we can't really see the cytoplasm, but we can see ribosomes and DNA kind of floating around inside that cytoplasm. There aren't any internal compartments, and so sometimes I describe these prokaryotic cells as like a swimming pool where there is just a bunch of, it's like the city swimming pool. Everybody's just kind of floating around next to one another. There's no separate compartments. You got DNA floating around right next to the ribosomes. What I'll say about this before we move to eukaryotic cells is that there's a lot going on in the same area. You've got DNA replication occurring right next to protein synthesis. So the DNA has to divide whenever the cell replicates, and then proteins are built on those ribosomes. Those are 2 different chemical processes, and right next to one another, when they have to happen right next to one another, it makes both of them less efficient. I'm trying to think on this campus if we have an example of it. Well, some campuses, they have these study halls where instead of just a big room and everybody gets together and study, it's sectioned off into these separate little rooms. Have you ever seen one of those? So that way, if you're studying a biology test, you can close the door and you don't have to listen to people studying history or whatever it is, because that doesn't pertain to your test. And you can more effectively study because you've blocked out all these other things that are not relevant to you. And in eukaryotic cells, those types of chemical reactions can take place. You have many different types of chemical reactions occurring because there are many different compartments that can create different conditions for these reactions to occur. In a prokaryotic cell, it's just one blob of cytoplasm. There are no separate compartments. And it's one of the reasons why DNA replication in prokaryotic cells have higher mutation rates than in eukaryotic cells. In eukaryotic cells, they have sectioned off all the DNA inside its own organelle, the nucleus. And now that DNA replication happens more efficiently with less mistakes because protein synthesis happens out here in the cytoplasm. They've separated those 2 processes in these complex cells. Turns out separating your DNA allows a bunch of other things to occur out here in the cytoplasm, like the fact that these eukaryotic cells can gain additional organelles. Before I go away from this picture, I want to point out that one of these organelles in a eukaryotic cell, the mitochondria. It's why I wanted to grab this slide. We can see the orange compartments here represent the mitochondria of an animal cell. And they are about the same size as a prokaryotic cell all by itself. And we see there's at least 5 lines of evidence that suggest that these organelles of our cells, these mitochondria, at one time were their own prokaryotic cells. And they formed a symbiotic relationship with cells where they could live inside the cytoplasm and not get consumed as a food source. Let's look at prokaryotic cells first. I mentioned that they are all small cells because they have to exist below this threshold where there is more surface area compared to the volume inside. We can see as cells get too large, there is not enough surface area for the proper amounts of nutrients to diffuse in and waste to diffuse out. This picture shows a comparison between plant cells and animal cells. I wanted to get a plant cell model and bring it in here, and I couldn't find one. We do have an animal cell, but I have a picture of a plant cell coming up next. In both plant and animal cells, we're going to see that the cells are smaller than you can see with the naked eye. But when we look at microscope slides, we're going to see plant cells are going to be larger than animal cells. And that puts them kind of up against that threshold where there's so much volume inside and not really enough surface area for things to get in and out easily. But plant cells have an adaptation to handle that, and it looks something like this. It's a large central vacuole that exists in the center of plant cells, and what it does is it kind of pushes all of the organelles in a plant cell, like these chloroplast and the nucleus, out to the very perimeter of the cell. I'm trying to think in here, if I took these tables and I arranged them so that there was like a big square in the middle and we had to walk around the edges of the table, we would bump into each other a lot more frequently because we couldn't use all the space in the middle. These central vacuoles kind of do that to a plant cell. They force these organelles around the perimeter, so they have to interact with one another. It also makes it easier for when things like oxygen diffuse in or carbon dioxide diffuse out. All of those organelles are right next to the membrane. So plant cells have this big central vacuole where animal cells up here clearly don't. Let me see if there's anything else that I know I've said this with words. Cells have to minimize their volume and maximize the surface area so things can get in and out easily. Here's a picture of the different cell types that we're going to introduce. And as I'm saying that, I feel like I remember in Unit 1, we did see a slide that introduced the 3 domains of life. It looked something like this, and I don't know why the colors aren't coming out very easily. But there should be kind of a reddish tint around this branch that separates bacteria from this, well, at least we can still see this color. There is a branch of archaea. Both of these 2 branches of life, archaea and bacteria, are made entirely of these prokaryotic cells. So we'll say bacteria and archaea. And then eukarya. 3 domains of life, 2 of them belong to these prokaryotic cells. And since they both are, I said we would point out the similarities and differences between these groups. They're both made out of prokaryotic cells, so they don't have organelles or any other internal compartments, but they're on separate branches for good reasons. When we look closely at their cell walls, we're going to see that each of these have different substances in their cell walls, and also how they package their DNA is a little bit different. So how these cells replicate is also a little bit different. Again, they have similarities that they're both simple cells, but they are not part of the same branch. They do have some distinct differences. And it turns out our group, this eukarya branch that we belong to, share a more recent common ancestor with archaea than we do with bacteria. Turns out bacteria, their DNA doesn't loop around these little proteins called histones. Our DNA does coil up around these little histone proteins. Archaea also has DNA that coils up around those little histone proteins. So we have things in common with archaea that we don't have, that neither one of those groups, archaea or eukarya, have in common with bacteria. Anyway, 2 of them made out of prokaryotic cells. I jumped over this slide, but we do have a list of the 4 similarities that all cells have, whether we're talking about prokaryotic cells or eukaryotic cells. Okay, so similarities we've got. When we look at prokaryotic cells, even under high magnification, they don't have any of those internal compartments. This comes from a lab that we're going to do probably in a couple of weeks when we hunt down prokaryotic cells. And this is a slide of yogurt. I just grabbed some random yogurt from the grocery store. We put it under the microscope, and you can see all of these prokaryotic cells. You know, it sounds, I used to get creeped out by that. I used to think, I don't want to eat yogurt. There's bacteria in there. But it's crazy to think that, you know, I heard somebody even talking the other day in class. They're like, does it creep you out to know that you've got like layers of bacteria that live on your eye, like your eyelids and things like that? And it sounds creepy, but it just is, you know, we have this sometimes too much of a self-centered view on everything, and instead each one of us is like an entire ecosystem of organisms. You've got all kinds of bacteria that cover your surface, also in your digestive tracts. They even sell that probiotic, whatever those drinks are, that can make sure that you have the proper amounts of gut bacteria. For example, like if you eat lettuce, you don't have any enzymes in your body that break down lettuce, but if you have the proper population of gut bacteria, one of those bacteria in your gut can break down that cellulose, and the byproducts are going to be sugar. Your body can absorb that sugar. In fact, there's been a big push, I think, in the last decade or so where they have really improved a lot of people's health by making sure that they have the proper amount of gut bacteria. Well, I don't even want to say this. We were just eating pizza earlier. What's that? Now I have to? Do you really want me to? Well, okay, let's do it. Nobody's eating pizza anymore, but there was something I was watching the other day, and you don't have to take my word for it. You can Google, well, don't Google this while you're here at school. But they have been doing fecal transplants. Has anybody ever heard of a fecal transplant? Yeah. They take the poop from somebody that's healthy, and they put it in somebody that is unhealthy. And that sounds crazy, but what they're really doing is that in your colon, that's where all the gut bacteria live. And if you have had a poor diet, or for some reason you have lost your gut bacteria, then one easy way to do that is to add the gut bacteria from somebody that is healthy. It is tough to think about, but it is something that's effective. Maybe something that is less gross to think about. It's been a few years since I've got to go to these physiology conferences, but I was always interested in mammals and how some mammals like rodents and bears can hibernate. You ever thought that was crazy? Like how can they just sleep for months and then not eat, not go to the bathroom, and then all of a sudden wake up and then move around? How can some mammals do that? And it turns out that the mammals that can successfully hibernate, before they hibernate, they're always eating a bunch of stuff. And what they're really doing there is they're loading their colon with the proper amounts of gut bacteria. And these gut bacteria, they break down, they start to feed on the lining of the intestines as well. Like they provide the food, the energy for these hibernating mammals over the winter. And if they don't successfully have the right amount of gut bacteria, then they will wake up from hibernation too early and likely not have enough to eat or freeze to death. So successful hibernating mammals have the proper amounts of gut bacteria. It just kind of points to be a successful organism, you've got to have the proper amount of a bunch of different types of cells, not just your own body cells. So anyway, bacteria are gross, but we need them to survive not only on our surface, but also in our colon. Let's move on. I was just talking about bacteria. These are prokaryotic cells. They are found in yogurt. We have them on our body surface, but there are also another group of prokaryotic cells that we don't find on the surface of our body or in yogurt. These are the extreme types of prokaryotic cells called archaea. And they're sometimes called extremophiles because they live in these extreme types of environments that normal bacteria couldn't survive in. There are these salt flats. This is an area where seawater has become landlocked. And as it evaporates, it just leaves behind higher and higher concentrations of salt water, and it becomes so salty that normal bacteria die off. But it provides an opportunity for these archaea to thrive. They can thrive in extremely salty environments, extremely low pH environments. This is limestone rock that's being eroded by some of the water, and the minerals that are dissolved from this calcium make the water extremely acidic. Normal bacteria couldn't survive there, but archaea can thrive. These are some of those hydrothermal pools that the water is so hot that it's practically boiling all of the time, and it prevents normal bacteria from being able to operate. Just like here is a methanogen. This is also a type of archaea that doesn't live in normal environments. It lives in oxygen-free environments. So like inside the digestive tracts of us or cattle, you find a lot of these methanogens. And again, they break down things like cellulose, or even nowadays you find them in industrial uses. Has anybody driven out towards like I forget what direction it is, but there are landfills between here and Hutto where they used to just dig a hole and drop your trash in the hole and cover it up. And instead what they've done, this kind of green revolution with trash, is that they dig a hole, they then line it with this thick layer of rubber, and then they dump all of your trash in there, and then they put a second layer of this rubber-like material over the top, and they seal it off. It's like a Ziploc bag that they will then get these tubes, and they punch it down through the lining, and they use it like an air pump, and they suck out all the air. So it's like a vacuum-sealed bag of trash under the ground. And now that we've vacuumed out all of the air, we've created this oxygen-free environment for extreme bacteria. Normal bacteria couldn't operate there, but these extreme bacteria now thrive that we've removed all the oxygen. And they break down the trash, and as a byproduct, they start to produce this methane gas. And those same pumps that were sucking out the oxygen originally can now be used to suck up that methane gas. And that's sent over to a power plant where it's used to heat up water and create steam and spin a turbine and generate electricity. So these methanogens are used in landfills to make methane gas that's being used as a power source. So they're not all bad. But we do need to know some characteristics about prokaryotic cells. And so far we've got lack of nucleus, lack of any other organelles. They're smaller than these eukaryotic cells. We can list at least two more, and let me find my color I was writing with. One of them is that all of the prokaryotic cells, they've got DNA. Yes, all cells have DNA, but the DNA in prokaryotic cells is in a circular loop. Circular loop of DNA. That's not what we're going to find in these more complex cells. In the complex cell, the DNA is broken up into chromosomes. I'm going to add a term to that. Instead of saying chromosomes, I'm going to say linear chromosomes. So the DNA is broken up into linear chromosomes where it's in kind of like a circular loop in these prokaryotic cells. This is a really long circular loop of DNA, so it's kind of hard to look at the whole thing from this one picture. But there are, in addition to the circular chromosome, there are these tiny loops of DNA called plasmids. There's one right there, and then there's another one over here, and we can even see a third one. A plasmid is, if this circular chromosome is like a big necklace, then those plasmids would just be a bracelet. They contain 2 or 3 genes, small loops of DNA. The fifth and final difference we can list between these simple and complex cells is if their DNA is different, then how the cell replicates is going to be different. We're going to see that prokaryotic cells are going to divide by what's called prokaryotic fission, or it's also called binary fission. So binary fission or prokaryotic fission is the way that these prokaryotic cells divide, versus eukaryotic cells are going to divide in 2 ways, either mitosis or meiosis. We'll say divide by mitosis. Now we're back to our comparison between prokaryotic and eukaryotic cells. We can see the nucleus, we can see other internal compartments. We're about to take a tour of those other internal compartments. And maybe first talk about how these eukaryotic cells arose in the first place. I was kind of making a big deal about this earlier. I was saying this is one of the biggest advances in evolution, the fact that these prokaryotic cells, instead of engulfing one another and competing against one another, they started to work together. This is a prokaryotic cell, but we can see some of the membranes have folded inward. So you've got these multiple layers of plasma membrane folded in on itself. This increases the amount of surface area, so there's more surface area along the membrane for things to move in and out. And it shows you that some of these prokaryotic cells have adapted their membranes to fold inward. And one of these prokaryotic cells adapted their membranes to fold inward and ended up surrounding all of the genetic material, all of the DNA in the cell, got sectioned off in its own little compartment. That particular compartment, the first organelle, we call the nucleus. And you can even see some of the residual membranes that were folding in to surround all of that DNA. They have left these, what look like a series of misshapen pancakes, all stacked together that surround the nucleus. We're going to refer to those series of membranes as the endoplasmic reticulum. We'll see a smooth and a rough version of this, but it surrounds the nucleus. It's made out of the same membranes that the nuclear envelope is made out of. When we look closely, after those membranes have sectioned off all of the DNA, now DNA replication can happen in its own little compartment, and other things can happen out here in the cytoplasm. For the first time, you get these other prokaryotic cells that get engulfed, but not broken down as a food source. They get a home, and this happened not only once, but it's happened at least 2 times, because plant cells have both mitochondria and chloroplast in them. This is also, I wish I had time to go on a tangent in this. It's interesting to think that we all come from a lineage of cells who have inherited mitochondria. So all of our cells look something like this, where we've got this organelle called the mitochondria that breaks down sugar and makes ATP for our cells. But plants, they also have mitochondria, but in addition to the mitochondria, they inherited another organelle that allows them to make their own sugar. So plants are self-sufficient. As long as they have water and sunlight, they have an organelle that can make its own sugars, this chloroplast. And then they also have the organelle that can break down sugars, the mitochondria. Anyway, not that you could argue in that sense that plant cells are more advanced than animal cells because they have organelles that we don't have. And they can do things that we can't do, like make our own sugars. Okay, so this process of prokaryotic cells working together is backed up by some evidence. So if we look inside these chloroplasts, or if you look inside these mitochondria, what's kind of fascinating is that they have their own DNA inside there. There's nothing inside of our nuclear DNA that has instructions to build these mitochondria. So where do they come from? Turns out we get all of our mitochondria from the cytoplasm of our mother's egg. You don't have any of your dad's mitochondria. So if you get your genome sequenced, they can sequence your nuclear DNA, or they can sequence your mitochondrial DNA. If you get your mitochondrial DNA sequence, it's only from your mother's side. If they want to trace, well, that's how they can trace different mother or father's lineage. If we look inside the mitochondria, or if we look inside these chloroplast in plant cells, we're going to see that both the chloroplast and the mitochondria, they have their own DNA sequences. See if I had a picture of that. This isn't clear enough that we can see inside this organelle, but there is a picture coming that we'll be able to see further inside this organelle, and you'll see that it has fluid inside there, like a cytoplasm. It has a membrane. It has its own ribosomes. It even has its own DNA sequences, and those DNA sequences are in circular loops, just like they would be in prokaryotic cells. Those organelles, I said, where do we get more mitochondria? They replicate on their own. They divide by way of binary fission, just like prokaryotic cells do. Later on in this class, we'll talk about cell cycle. Cells have a life cycle, just like we have a life cycle. And whenever a cell gets towards the end of its life cycle, the nucleus is going to divide, and that is the signal that tells all of these other organelles. When the nucleus is getting ready to divide, these organelles go into their divisions. That means that these mitochondria are constantly being born and are dying inside our cells. And you could have a cell that's littered with the carcasses of a bunch of dead mitochondria. Luckily, there's an organelle that will remove any debris or old, worn-out organelles inside of the cell. So this is just showing us the evidence that this process of endosymbiosis occurred. Endosymbiosis, we said, is the name given to this symbiotic relationship between multiple prokaryotic cells, where one started to live inside the other. We have now built the beginnings of our first complex cell with different internal compartments and different chemical reactions taking place. So primary endosymbiosis is when the first prokaryotic cell went inside of its host eukaryotic cell. So there is a nucleus. We have some organelles that have been taken on board. In the second half of this class, we'll learn how this led to green algae down here and red algae up there. We will later see that these red algae and green algae also get engulfed by other eukaryotic cells. And this leads to what looks like one of those like nested series of dolls. Have you ever seen one of those nested series of dolls? Some of these larger complex protists are like that, where you have these large eukaryotic cells that have a bunch of internal compartments that each might have their own DNA. We have cells that have their own compartments with their own DNA, and those would be the mitochondria. We'll look at all these different diverse protists. I think we get to them. That's still the second half of this class. So I'll hold off on that. I'll just show you this picture to plug that there is a variety of different microorganisms. Anything that is made out of eukaryotic cells belongs to this branch of the eukarya. We are on this branch. Animals, fungus, and plants are somewhere along the group of eukarya. But there's a bunch of other varieties that don't look anything like plants, animals, or fungus. We get to tour them next time. I'll jump away from prokaryotes to look at eukaryotes now that we've listed similarities between the different types of cells and differences between prokaryotes and eukaryotes. Let's look just specifically at these eukaryotic cells. And it looks like we're starting with the plant cell. I'm going to compare, in terms of eukaryotes, we're going to compare plant cells to animal cells. Plants and animals are both made out of these eukaryotic cells, and we should be able to list, well, there are 4 similarities between them. I'll do the similarities, and you see if you can point out 4 differences that are visible between this plant cell and the animal cell if we go back and forth between them. The 4 things that they all have in common, remember there's the plasma membrane, then there is the fluid inside the cell that everything is floating around in, that's the cytoplasm. We've got the DNA, there's some DNA, and then also ribosomes. We can see those little dots that represent ribosomes. So 4 things that all cells have in common. Maybe like that Sesame Street game, there's the 4 things that are not present on animal cells, but we can see them right there on the plant cell. Let's see how much coffee I can drink before y'all can point out those 4 things on a plant cell. So I'm going to see how much coffee I can drink before y'all can point out 4 things that you can see there on a plant cell that would not be present on an animal cell. Chloroplast is one of them. The green-colored organelle is something that you only find in plants. We said that was one of the advantages that plant cells have over us because they can make their own sugar. Chloroplast is one of them. Plant cells are larger than animal cells, and that would make it difficult for these organelles to interact. But they have a way to get around that. That large central vacuole kind of forces the organelles out to the perimeter, and it makes them interact with one another. Central vacuole, chloroplast, that's 2 out of the 4. I might be able to kill this. We got 2 more left. Still kind of hot. Cell wall. That's right. So there's a rigid cell wall that we find in plant cells that we don't have in animal cells. And the fourth one kind of ties into that cell wall. There is a special type of cell junction called a plasmodesmata. It's unique to plant cells because this type of cell junction crosses a cell It crosses a cell wall. So if you have a cell wall, you also have these special types of cell junctions called plasmodesmata. You can see it looks like a hollow straw that just connects the cytoplasm of two adjacent plant cells. It allows for what's called cytoplasmic streaming. We'll see this under the microscopes later. It's where cytoplasm just flows from one cell to the next adjacent cell through these little openings called plasmodesmata. So good. Those are the four things plant cells have that animal cells don't. When we look at animal cells, there's this thing that maybe sticks out at you. This is a flagella. We're going to see that animal cells sometimes have a flagella. They don't always have a flagella. But this isn't something that's unique to animals. If I can go back to plants for a second, this plant cell does not have a flagella. But I'm diverging just a second to talk about plants next semester. When we talk about plant groups, we're going to see four major plant groups. And the first two of those plant groups, mosses and ferns, they have flagellated sperm. So they're plants that produce cells that have flagella. And these flagella have to swim through a droplet of water to find an egg and complete fertilization. Plants that figure out how to break that link with water, the first conifers, have figured out a way to get sperm to egg by using wind as opposed to water. So some plants are going to have flagella. This isn't unique to animals. So that doesn't count. But we did list the four things that plants have. Let's look at some things that we find on the animal cell. And maybe the first organelle, if we're going to look at any of them, is this large nucleus. It's kind of everything is based around this nucleus. So when we look at it, you can see inside the nucleus there is that dark region called the nucleolus. Maybe write down the nucleolus is where all of the RNA comes from. This is maybe a little review from last lecture. We talked about RNA and DNA. Both were types of nucleic acids. They stored information. But DNA had some differences that RNA didn't have to deal with. We said DNA tended to be double-stranded. Most notably here, DNA is going to be locked inside the nucleus and can never leave. Whereas RNA is going to be able to move out of the nucleus and operate out here in the cytoplasm. And we'll talk about this a lot, we're going to talk about different types of RNA floating around out here in the cytoplasm and building proteins. And if you ever think, where did this RNA come from? It comes from right there. It comes from the nucleolus. So all DNA and all RNA come from the nucleus, but only RNA can leave the nucleus and operate out in the cytoplasm. DNA stays inside the nucleus. Okay, we can see that nuclear envelope. Maybe I can zoom into that nuclear envelope. Well, at least we can. Here's a picture where we've zoomed into that nuclear envelope. It's a double-layered plasma membrane. It's folded twice, and kind of embedded in that membrane are these what are called nuclear pores. It's a protein channel, and it's just big enough to allow ribosomes to escape the nucleus and make their way out into the cytoplasm. The other thing that you can see up here, in addition to the nucleolus, there is all of this other stuff out in the nucleus called chromatin. Think of chromatin as DNA in its fluid state. I said cells have a life cycle just like we have a life cycle. And for a majority of a cell's life cycle, when a cell is just being a cell, all of the DNA is unwound, it's unraveled, it's in its fluid state called chromatin. And that allows certain segments of DNA to be exposed. So if you were an enzyme and wanted to read that section like a recipe to build a protein, it's exposed for you to do that. Cells are only going to condense all of this DNA into chromosomes when they get ready to divide. And then the genes are locked up. They're not readable at that point. My brain's always thinking in weird analogies, and I used to have this recipe book that my great-grandmother had written some things down in, and I can't remember those, but if I have the recipe book in front of me, I can read it and follow it. But one time I had to move, and I put it, it's an important book, so I put it in this really strong box, and then I put it in the storage unit somewhere, and it's packed away, and when it's condensed like that and packed away, I don't have access to it. I can't read it. I'm describing here somewhat of what we're going to introduce later as epigenetics, where you don't make changes to the DNA. We haven't changed a nucleotide in the DNA sequence. Instead, we've changed how that DNA packages up. You can turn off genes by covering them up, preventing enzymes from being able to attach to that part of the DNA. We'll talk about how this DNA packages up and exposes genes or covers them up as we move forward. But DNA is, when it is in its fluid state, we refer to it as chromatin. I'll zoom out of the nucleus for a second, and we can look at this endomembrane system that surrounds the nucleus. Parts of this endomembrane system are covered in ribosomes. This is what we call the rough endoplasmic reticulum. And then if you look way over there, there's a little bit of this endoplasmic reticulum that lacks ribosomes. We need to know the function of the rough ER and the function of the smooth ER. And the presence of ribosomes tells us something about the function. When we see one of those ribosomes, we know that proteins are being built there. So maybe you can write on this one the function of those ribosomes are to build proteins, the assembly sites for proteins. So when we look at this rough endoplasmic reticulum, its function is to package proteins that are being built. Here is a ribosome on the rough endoplasmic reticulum. You can see it has built a chain of amino acids, and that chain of amino acids is folding up into its 3D shape. So packaging proteins and then shipping them off to different parts of the cell is something that happens in the rough endoplasmic reticulum. Over there is the smooth endoplasmic reticulum, and since it lacks ribosomes, that is a clue to its function. All endoplasmic reticulum packages stuff that the cell makes. But since there are no ribosomes there, we know that it's not packaging proteins. It's going to be packaging either carbohydrates or lipids. The cell doesn't package nucleic acids. That is an information storage molecule, but the cell can build carbohydrates, it can build lipids, and package those up and ship them off in smooth ER. But if it's building proteins, that's going to be packaged up in the rough ER. Here's that rough ER. We can see some of the proteins have been built. The purple circles there represent the proteins that are being built. And here you can see some of those proteins that leave the rough endoplasmic reticulum are making their way to another organelle called the Golgi body or Golgi apparatus. This Golgi apparatus, it looks like a series of stacked pancakes again, but it has one side that receives packages called the cis face of the Golgi body, and then there's a trans face of the Golgi body that it's like the out box. It sends packages away. But if you see what's happening, not that this is visually obvious from looking at it, but the cis face or the cis side of this Golgi body is going to receive proteins that are not yet fully functional. They haven't been modified to their final structure yet. We know changing the structure of something changes how it functions. So as these proteins make their way through the Golgi body and get their structures altered, they're going to have different functions when they come out of the other side. Look at how some of these proteins are functioning as insulin. This is a chemical signal that's produced by one cell, leaves the cell, goes out into the bloodstream, and communicates to other parts of the body. That's a protein-based hormone. There are other protein-based structures. These are digestive enzymes that will digest worn-out parts of the cell. Maybe an analogy is better. So since we're thinking about this Golgi body, I was driving down the road, I guess it's been a while, but has anybody ever seen one of these 18-wheelers driving down the road that's got like a flat trailer on it, and it's carrying a gigantic bar of aluminum? There was an aluminum power plant or an aluminum plant in Rockdale that closed down a couple of years ago. But they used to mine aluminum, and then they would melt these big bars of aluminum down, and they would put them on the back of an 18-wheeler, and they would ship this big bar of aluminum. You would sometimes see it driving down the highway. And what they're doing, they're basically doing the same thing that a Golgi body is doing. They're taking these unfinished materials and repurposing them for different things. And I would see that bar of aluminum on the back of the 18-wheeler driving down the road, and it would be going to an aluminum processing plant where they could take that bar of aluminum and maybe reshape it into, I'm looking around the room for a Coke can. They could shape it into some type of can that you could put your drinks in. Theirs looks like something that's made out of aluminum back there. I'm looking at, yep. So that's something that it got modified into its final function. Or we could modify those aluminum sheets into fence material. Or we could make a fancy rim for your car. There's all kind of things we could mold that aluminum into. So originally it's in its unfinished state. Proteins are the same way. The Golgi body takes unfinished proteins and then repurposes them for a bunch of different reasons. Like that aluminum bar can be modified into a bunch of different products. So again, cis face receives unfinished proteins, and then they come out of the trans face fully functioning as different types of proteins. So modifies unfinished proteins. Here are some of those unfinished proteins. A lysosome is one of these proteins that comes off of a Golgi body that goes to work inside the cell by digesting either food that's been engulfed by a cell or by digesting worn-out organelles like this mitochondria. So those parts are recycled and used to make more mitochondria. We said vacuoles are something that's unique to plant cells. This is a black and white picture, but you can still see that this large central vacuole has forced those chloroplasts. We can see 3 chloroplasts and 1 nucleus out to the very edges of the cells. And maybe a decent segue for what we're about to see in lab in a second. That is, we did a microscope lab already, and we saw that there are microorganisms in pond water. This is not one of the microorganisms that we happened to find the other day. I thought we could have seen it, maybe in another sample of pond water. But if you're a microorganism swimming through pond water, sometimes you run into a part of the pond that is hypotonic, and you could end up swelling and taking on too much water. As an animal cell, we might have mentioned this last time, it's problematic to take on too much water because there's no rigid cell wall that prevents us from rupturing. So animal cells can survive shriveling up better than they can survive taking on too much water. These single-celled organisms have ways of preventing taking on too much water. Those organelles are called contractile vacuoles, and when this cell swims into a hypotonic solution and starts to take on too much water, these organelles fill up with that water and then are capable of contracting and forcing a large amount of water directly out of the cell. We'll see a video of this next time. I am in a different room, so I don't have it set up currently. But let's maybe let this be a review slide, and I want to draw one more thing on the board once we get past this. It's just to set up our lab results that we're about to see. So for today at least, we got through some functions of organelles. We compared similarities between all the different types of cells, whether it's prokaryotes or eukaryotes. We know 4 things all cells have in common. If we don't, maybe you should write this down a few times before the next exam. It's likely to be somewhere on there. We should know at least 4 differences between prokaryotes and eukaryotes. And then we should know some differences between plant and animal cells. We listed 4 differences between plant and animal cells. After all the similarities and differences between the cells, we looked at some particular organelles that you would find in the eukaryote, like the nucleus, like the endomembrane system, both smooth ER and rough ER. Smooth endoplasmic reticulum, the function we know is packaging carbohydrates and lipids. Rough endoplasmic reticulum is packaging proteins. Golgi apparatus is receiving packages of unfinished proteins and then repurposing them for a variety of different reasons. So what goes out of the out box, the other side of the Golgi apparatus, are fully functional proteins. Lysosomes and vacuoles are examples of some of those structures that the cells produce. Lysosomes, we said, are digestive enzymes, and vacuoles are those large structures inside plant cells that force organelles out to the perimeter. We're going to come back and we're going to look at mitochondria and chloroplast in more detail next time. Here's that picture that I promised that shows an even closer zoom on a mitochondria. You can see those 4 things that all cells have in common, even that mitochondria. It's got a circular loop of DNA, it's got ribosomes, it's got it's got cytoplasm, it's got fluid inside this cell. I almost said chloroplast. Cytoplasm, ribosomes, DNA, and a plasma membrane. Here is the chloroplast. It too has those little circular loops of DNA. It has its own ribosomes. It has its own membrane, fluid inside that membrane. We'll pick up with the cytoskeletal elements, I guess, on whatever the next day is, Tuesday. But I'm going to turn the screen off for a second and ask a couple of questions about membrane transport. So measures the solute concentration of a solution. Solutes, remember we did this before. I keep using the example of tea and sugar as my solutes, as the stuff that we would dissolve in the solvent, which is water. What am I doing? Together we said this makes up the solution, which in this case is just some sweet tea. And I guess if you drink a lot of sweet tea like I do, you've been places where they make sweet tea and it's got just a ton of sugar in it. It's so sweet, it's sometimes you can feel that it's thick. It's just got so much sugar in it. And then other times you'll go and you'll get a glass of sweet tea that doesn't have as much sugar in it. Right? The amount of sugar, the amount of solutes that are floating around in this solution, we can put a number to that. One that has less solutes in it, the number is going to be lower. Maybe this is only 100, again, osmoles. Osmolarity is allowing us to put a number on the solute concentration. It doesn't tell us what those solutes are, whether they're tea, whether they're sugar. They're just particles floating around in a water. So if there are more solutes or more particles floating around in a solution, that's going to get a higher number. Maybe this one's at 400 milliosmoles. Right, so osmolarity is just a number that describes how many particles are floating around in this solution. We've got that so far, right? We did this last time. So then let's compare osmolarity to this term tonicity. Tonicity is also referring to solute concentrations, but specifically tonicity we can describe as it refers to the solute concentration of extracellular fluid. Let's unpack that. Refers to solute concentration of extracellular fluid. Okay, now we've got that up there. And the terms that we use to refer to that extracellular fluid, we can refer to it as being hypotonic, and we can describe these terms. We know hypo is always below, and tonic or tonicity is telling us solute concentration of the extracellular fluid. Hypertonic is telling us if this extracellular fluid is above or higher than the solute concentration of the extracellular fluid. Isotonic is just telling us the same, and then tonicity is telling us solute concentration of extracellular fluid. We built each of these solutions in lab last time, and I'll try and draw one of those real quick. We had, I think we can fit them in here. Let's put, I'll use this color. We didn't have numbers on them, but I'm going to label this one as beaker number 1. Let's label this beaker number 2, and then we had our third and final beaker. And you'll remember each one of those had what we called a fill line, and that fill line measured 300 milliliters of fluid. So 300 milliliters were found in each one of these cups. In the first cup, we said all 300 milliliters were water. So we filled it up with 100% water, and there wasn't any glucose. It got 0% glucose. So whenever we made this was our first beaker. It was easy to do. We just filled it up with water. But the next two had a different amount of water in it. The second one had 200 milliliters of water, and the third one had half of that. It had only 100 milliliters of water. And then what you had to do is you had to add the corn syrup to fill the rest of this volume of the cup all the way up to the fill line. So this got 30% glucose, and the rest of it was water, so that whenever we mixed this stuff together, you had some sugary water, about 30% glucose. This one we filled with 70% glucose because it had just a very little bit of water in it, and you had to stir this one pretty good to get it all mixed up. But once you mixed it all up, we had a solution that was much more sugary, and you can feel that it was even thicker. So that just indicates a higher concentration of solutes. Right, we all did this one. So now we've got our 3 different solutions. And we can put a number to that. This has no solutes floating in it. So if we wanted to put a measure of osmolarity, this would have 0 milliosmoles. It's got no particles floating in it whatsoever. This one we have about 30% glucose. Let's put a number to that. I'm going to say, we'll say 300 milliosmoles. So that gives us a number to the solute concentration. There's more solutes dissolved over here, so let's say this is 700 milliosmoles. Are we doing good so far? The one with the more solutes has the higher osmolarity reading. Here comes our first question. We just talked about solutions that are hypotonic, hypertonic, or isotonic. Is this solution, could we call this solution either one of those? Is it hypotonic, hypertonic, or isotonic? Just this beaker all by itself, as it is right now on the board? Why is it not hypertonic? We don't have anything to compare it to. It's just all by itself. But remember what we did in lab. We have those eggs. There are eggs that represented human cells, and we said each egg, like a normal human cell, has a certain amount of solutes floating around in it. Some of these are plasma proteins, other ones are sodium and potassium ions, you've got glucose, you've got a bunch of particles floating around in there, and we can put a measurement to how many particles there are inside a cell. It's about 300 milliosmoles. So our egg represented our body cell, and we submerged an egg into the first beaker. So what you have in there is an egg that has 300 milliosmoles. Again, that represents a certain amount of solutes floating around in it. Now that we have an egg in here, we can refer to this extracellular fluid. This stuff surrounding the cell is our ECF. If we're thinking about that ECF, how would we describe it now that we have a cell to compare it to? Is there more or less, or is there the same amount of solutes in this beaker versus if we compared it to inside that egg? There's less. There's no solutes out here. That's right. So what term would we use? Hypotonic. This is another clue. So hypotonic. Remember that when cells are put into a hypotonic solution, the cell will swell. And there was a guy in my class yesterday that says he always remembers that a cell is going to swell up when you put it into a hypotonic solution because of that O right there in hypotonic. It looks like a swollen cell that's been put into a hypotonic solution. It swells into this spherical shape. The thing that I want you to remember is that water molecules are always attracted to solutes, to glucose, to sodium, to plasma proteins. Sophia, if you were to get up and just start to walk around the room and you were acting as a glucose molecule, water molecules, whoever was acting as a water molecule would start to follow wherever you go, right? Solutes have this ability to pull on water molecules. And like you just said, there's more solutes inside the cell. So what happens is water molecules are drawn towards those solutes. When a cell is in a hypotonic solution, water goes into the cell and the cell is going to swell. Now let's jump over to this one. This is our, all by itself, we can't say whether it's hypotonic, hypertonic, or isotonic. But as soon as we drop our cell in there, again, the cell is 300 milliosmoles, so there's a few solutes in it, but there's way more solutes around the outside of the cell. So now we can refer to this ECF as hypertonic. Hyper is just above the amount of solute that you would find inside the cell. So whenever cells are put into a hypertonic solution, they're going to shrink. Cells shrink, or they crenate, whenever they're put into a hypertonic solution. And the same thing is true if you think about that R. The way that R looks in hypertonic, it kind of looks like a cell that has crenated or shriveled up a little bit. It's not a perfectly round cell. So maybe that's one way that you can remember cells swell in a hypotonic solution. And then in a hypertonic solution, the same thing. Water is pulled to the side of the membrane wherever there are more solutes. So in this case, water is pulled outside of the cell. Finally, if we look at our isotonic solution, we've got about the same amount of solutes inside the cell as we have outside the cell. So some water is going to make its way in, but at about the same pace, some water is going to be pulled out. So there is no net movement of water in an isotonic solution. So isotonic, we said, was just the same tonicity, so the solute concentration of the extracellular fluid is about the same as the concentration of solutes inside the cell. Does that make sense? So now we know when to use hypertonic or hypotonic, and we know what to expect when we go in lab in just a second and look at our hypertonic beaker. We expect that cell to have lost water. We expect our cell that's put in a hypotonic solution to be bursting almost because it's so swollen with water. And then we should have a cell that is in between those two extremes that is as close as we could get to an isotonic solution.